The overall goal of this procedure is to monitor the events of mouse embryo turning, neuraxis elongation, and neural tube closure in an extrauterine environment in which conditions can be defined. This is accomplished by first removing the uterine horns from the anesthetized pregnant mouse at gestational or embryonic day 8.5. Next, individual mouse embryos are removed from the uterus with their yolk sac intact and much of the decidua. Then, the extra-embryonic decidua tissue is trimmed away, leaving the ectoplacental cone intact. The final step of the procedure is to transfer embryos in their yolk sac to roller bottles containing media with or without pharmacologic agents being tested and culture embryos for 18 to 24 hours in an incubator with continuous gassing. Ultimately, results can be obtained that show progressive addition of somites, apposition of neural folds, and closure with turning of the embryo into the familiar C curvature of the fetal position.
This method can help answer key questions in the field, such as what molecular pathways are involved in normal neural tube closure, what agents or supplements can help promote normal neurulation, especially in embryos that carry genetic mutations that predispose them to neural tube defects or NTDs.
Visual demonstration of this technique is critical, as the steps in the dissection can be very challenging. The embryos are small and can be easily damaged, and damage to the yolk sac can impair somite addition, turning, and also development of the circulatory system, preventing a successful outcome.
To begin, prepare culture medium by thawing male rat serum at 37 degrees Celsius. Next, heat and activate the rat serum for 30 minutes at 55 degrees Celsius, then spin it down at 10,000 RPM for 5 minutes at room temperature. Remove the supernatant and mix it in a 1:1 ratio with phenol red-free DMEM with high glucose. Sterilize the medium using a 0.45 micron syringe filter. Isolate the uterus and place it in a petri dish with saline. To remove the embryos, first rinse the uterus with saline to remove any excess blood and trim away any excess fat from the outside. Transfer it to a clean dish with room temperature thyroid saline to improve visualization. Under a stereo microscope, with hands stabilized on the stage, use small scissors or fine forceps to peel the uterus apart and separate each embryo. Insert fine forceps into the space between the uterine wall and the decidua and peel back the uterine wall using minimal tension to avoid rupturing the yolk sac. Next, orient the decidua so that the placenta is facing away from you. Then make an incision with the forceps along the line separating the dark portion of the decidua from the lighter portion, being careful not to cut too deeply. Complete the separation of the placenta from the top of the yolk sac, being careful not to tear the ectoplacental cone, or EPC. At this point, the Reichert's membrane on the yolk sac should be visible.
Removing the Reichert's membrane from the yolk sac is the most difficult part of the procedure, and it requires patience and a steady hand in order to delicately remove the Reichert's without damaging the underlying yolk sac.
Continue to remove the entire decidua, being careful not to pierce the yolk sac. Gently pinch and remove the Reichert's membrane by peeling it away from the EPC. If the EPC is damaged or separated from the yolk sac, embryos will fail to turn normally from E8.5 to E9.5. Once the Reichert's membrane is removed, the embryo should be easily visible through the yolk sac. Stage the embryo by counting the somites along one side, starting beneath the otic vesicle and ending at the tail. With scissors, cut the end of a plastic transfer pipette to use on the embryo without damaging the yolk sac. Fill clean roller bottles with 1 milliliter of medium per embryo. Depending on the type of roller bottles used, a maximum of 3 to 6 embryos can be loaded in each bottle. Using the cut pipette, transfer the embryos to the media-filled roller bottle, carrying over as little thyroids as possible so as not to dilute the medium. Attach the rubber plug to the top of the glass roller bottle. Insert the roller bottles into the roller culture drum so that a tight seal is formed between the plug and the drum. Once all the bottles have been attached, seal any empty slots in the drum with rubber stoppers, then activate the roller. Next, open the premixed CO2-O2 tank and adjust it to approximately 2 psi so that the outlet valve is releasing 1 bubble per second. For E8.5 E10 embryos, 20% O2 and 5% CO2 should be used. Ensure that the incubator is set to 37 degrees Celsius and cover or close the incubator so that the embryos are shielded from light. To assess embryo development, check them periodically for the addition of somites, the progression of turning, and the extent of neural tube closure. After 2 to 3 hours in culture, if desired, add pharmacological inhibitors or other treatments. When the incubation is complete, turn off the gas, stop the roller, and remove the bottles from the incubator. Transfer the embryos back to tyroids or PBS in a petri dish. Under the stereo microscope, the yolk sac should appear balloon-like, a heartbeat should be visible, and the circulation should be evident. Remove the yolk sac from the embryo by cutting a hole near the EPC and flipping the yolk sac and amnion over the head and rump of the embryo. Cut the umbilical cord to separate the yolk sac from the embryo. Finally, examine the embryo for somite number and neural tube defects such as open cranial folds, incomplete closure of the face, and/or an open caudal neuropore. Embryos can be staged according to the Tyler classification of multiple morphogenetic changes. The appearance of embryos pre- and post-roller culture is illustrated here. At the time of dissection, embryos should be in an unturned configuration where the tail is behind the head folds. After 24 hours in culture, embryos should have completed turning so that they are in a C-curved fetal position where the tail is in front of the head. This image shows, in the top row, the development of embryos without inhibitors added. The bottom row shows the effect of pharmacological manipulation with the Rho A kinase inhibitor Y27632, a known inhibitor of convergent extension during neurulation. This inhibitor results in a shortening of the embryos along the rostral caudal axis and inhibits cranial neural fold closure. Our data show that increasing doses of Y27632 progressively impair cranial fold closure and shorten the body axis, consistent with the role of downstream Rho A signaling in cranial neurulation and convergent extension.
Once mastered, the dissection technique can be completed in 60 to 90 minutes for a litter of 6 to 12 embryos.
After watching this video, you should have a good understanding of the steps required to culture gestation day 8.5 to 10 mouse embryos for the purpose of studying developmental mechanisms at a systems level.
Method importance. The ability to separate maternal, intrauterine factors from those that are intrinsic to the embryo during its development is an important tool for studying all stages of embryogenesis. The method presented here can also be applied to studies of other molecular pathways in development, allowing the direct manipulation of cellular function in embryos using a variety of reagents. Thank you for watching. Subscribe if it is useful.